Good news, everyone. Biology for Beginners is back once again. As always, I'm your host, Morgan, and this week we're going to be talking all about mitosis, which is a subject that I'm pretty jazzed about, so excuse me if I get excited. Also, excuse the angry duck noises in the background. I have accidentally disturbed their mid-afternoon nap, and they think I'm the worst thing in the world right now. Also, I have no freeze-dried mealworms for them, and that's clearly a sin. As always, at this moment, we are popping up those terms and definitions slides for you guys, so you can pause and grab the vocabulary necessary to understand what I am talking about today. Also, you can pop on over to my Patreon. The link for that is down in the description below, and grab my entire PDF slideshow for you to help study, to help keep on track, whatever you would like to do with it. Also, if you are a patron, you do get the Bio for Beginners and all the study material at least a day early, as well as some other early access to videos, but otherwise all of the study material, as soon as this video goes public, is absolutely 100% free. Before we start talking about mitosis in depth, let's talk about the cell cycle. So the cell cycle is basically the life cycle of a eukaryotic cell. And this particular cycle can be broken down into two main phases, interphase and the mitotic phase. And then both of those phases can be further broken down into their particular parts. Interphase is the phase that takes up the majority of the cell cycle. And this is the phase where there's a high amount of metabolic activity, and this is also all the quote-unquote normal cell activity will take place. The first part of interphase is going to be G1 or gap 1. And so this is where growth and just normal activity kind of happens. After G1, you get into S phase. Now, S phase is where DNA is synthesized, so it's the DNA synthesis phase. So you may be thinking, wait, shouldn't the cell already have DNA? And that is correct. Specifically, when we're talking about DNA synthesis, we're talking about when the DNA is replicated. So the DNA that's already hanging out inside that cell is going to be replicated and then synthesized into a copy of the original strand there. And S phase takes up about half of the whole interphase portion of the cell cycle. And then once the cell gets out of S phase, it moves into G2 or gap 2. And that's kind of where the final preparation for cell division happens. Now, G1 and G2 are the gap phases, but keep in mind they are not inactive. The cell is still doing plenty of cell things during those phases. That's just kind of what they're called because they're the gaps between mitosis and DNA synthesis. The other part of the cell cycle is the mitotic phase, or M phase as some places call it. I've never really heard it referred to that way in earnest. I've just had textbooks reference it as M phase as sort of an abbreviation. But the mitotic phase is only about 10% of the overall cell cycle. And this is the phase where cell division actually takes place. So the mitotic phase is made up of mitosis, which is where the cellular components are duplicated and distributed in the two ends of the cell before cytokinesis happens, which is when the cytoplasm actually divides and the two daughter cells are actually produced. And then once this cytokinesis happens, once the cell is done with the mitotic phase, both of those brand new daughter cells will then move into G1 and the whole thing will start again. If you'll notice at this point in our discussion of cell division in mitosis, this whole process seems a lot more complicated than it was in our previous video when we were talking about simple cell division and binary fission. Well, that's because the organisms we were talking about last week were prokaryotes, so they're much more simple organisms. This week, we've moved into the eukaryotic cells, and if we remember correctly, eukaryotic cells are much more complex than prokaryotic cells. And because of this, the entire cycle of dividing and creating more cells is going to be more complex as well. And just like the rest of the cell being more complex, eukaryotic chromosomes are much more complex as well. And not only are these chromosomes more complex, there are more of them inside the eukaryotic cell. So if we remember last week, or last video, I don't know if you watched it last week, but that's okay, I forgive you. The prokaryotic cell has a single looped strand of DNA that will carry all of the genetic information for that organism. The eukaryotic cell also has chromosomes that carry the DNA with all the genetic information for those organisms. However, their chromosomes are not a loop, and there's also many, many more chromosomes in each cell to carry all the necessary genetic components for that complex life. And so as a quick little example here of one cell carrying many chromosomes, we have humans have 46 chromosomes, dogs 78, hedgehogs will have 90, and each chromosome is going to contain that one strand of DNA per chromosome, and each strand of DNA may have hundreds or even thousands of genes on them. And that's carrying all of that genetic information that makes that organism that particular organism. And then also within each chromosome, you're going to get proteins that help maintain structure and control the gene activity. When you put the DNA and those proteins together, you get what is known as chromatin. While I'm sure we're all used to seeing chromosomes represented as that lovely little X, and they do indeed condense down into that little X, like we saw on the previous slide. That is an actual photograph taken from a microscope of a chromosome. Chromatin is usually just kind of hanging out in the nucleus of the cell in a bunch of long, thin, uncoiled fibers. And these fibers are much too thin to see under a light microscope. So you only really get to see the chromosomes when they are condensed, when the cell is getting ready to divide. And during division, that chromatin, as I said, will be coiled up and it will form those chromosomes that we are much more used to seeing. And then the chromosomes are going to be duplicated. This DNA duplication happens before everything coils, but as we talk about it, it's usually more talked about something happening at once. But it is important to remember that the DNA is replicated before the coils form. Because remember, in that S phase is where the DNA is replicated and the new DNA is synthesized. So the chromosomes that we see as the X are actually two individual sister chromatids, and each sister chromatid is a copy of each other. One half of each X chromosome is what each cell needs. So when those sister chromatids eventually split up, half of the X goes to each new cell. And the sister chromatids are going to be joined at that kind of pinched in sort of waist-like center portion known as the centromere. So to kind of summarize that again, normally chromatin is going to be a bunch of kind of long thin fibers that cannot be seen under a normal light microscope, but once DNA is synthesized and things begin to coil up and condense, you end up with these X-shaped chromosomes. Each half of the X-shaped chromosome is a sister chromatid that carries all of the genetic information for that particular chromosome that each new cell is going to need. And those two sister chromatids are joined at what is known as the centromere. Moving into the actual phases that will result in the two brand new daughter cells, let's begin with interphase. Now, remember, interphase is the phase where there's all that normal cell function. So it's just kind of the cell going out its everyday life. And during the large part kind of in the middle of interphase, you get that replication of the DNA, which happens inside the nucleus. And it's going to be in that loose chromatin form, not in the condensed little X-shaped chromosome form. And then by late interphase, so that end of that kind of G2 phase portion of it, the cell will have doubled most of its contents, and there will be 2 centrosomes, which are kind of those little yellow tubey things you can see in this diagram on the slide here. And then once the cell finishes up with that interphase, it's going to begin the process of mitosis. And to begin mitosis, we need to start at prophase. So prophase has things happening both in the nucleus and in the cytoplasm. In the nucleus, we're going to see those chromatin fibers begin to coil and fold and form those neat little chromosomes that have those 2 sister chromatids joined at the centromere. While in the cytoplasm, we're going to see mitotic spindles begin to form. These are just kind of these microtubules, and they're also known as spindle fibers. I know the book I'm using usually calls them spindle microtubules, but I'm more comfortable saying spindle fibers since that's what I've always learned, and that's usually what they're called in the biology class, so that's what we're going to be saying for the rest of the video. And so they grow out of those centrosomes. And then the centrosomes will begin to move away from one another towards the opposite ends of the cell at the 2 poles. Moving on from prophase, the cell will then enter the prometaphase. Now, originally, the parts of prometaphase were kind of split up into prophase and metaphase, but now it is recognized as its own individual phase of mitosis. So, prometaphase, because it's between the 2 more recognized phases. And what happens in prometaphase is that nuclear envelope begins to break down. So that membrane around all the chromosomes kind of disappears. And when that happens, we also have these centromeres reaching the 2 poles, so the opposite ends of that cell, and then spindle fibers begin to reach into the nuclear region. Now that nuclear envelope is gone, they can actually reach into the region and begin to attach to the sister chromatids at the kinetochore. Sorry, I'm really bad at pronouncing that particular word, but it's on the slide, so hopefully we can follow along that way. And once everything is kind of connected, almost in the correct place, the cell reaches the metaphase. And at metaphase, those mitotic spindles are fully formed and the poles are completely on the opposite ends of the cell. And with those spindle fibers, the chromosomes are kind of lined up at the metaphase plate. The metaphase plate is just kind of an imaginary plate directly through the center of the cell where those chromosomes line up. The middle parts, the centromere, is going to be lined up on it. And then each sister chromatid at this point will also be attached to a spindle fiber at the kinetochore. Again, I apologize for pronunciation. Metaphase is actually a very brief portion of the whole mitosis process because the chromosomes are only going to be in the middle for a very, very short period of time before the cell reaches anaphase. And during anaphase, the centromeres of the chromosomes are going to break apart, and then motor proteins in those kinetochores are going to walk the daughter chromosomes along the fibers towards the opposite poles. So each half of each chromosome is going to be pulled in a different direction. And then also those spindle fibers attached to those chromosomes are going to begin to shorten, while any spindle fibers that are not attached to chromosomes lengthen. And this whole process moves those poles even further away from each other, and the cell elongates some more. And then once the cell is pretty much through with the anaphase, it's going to reach the telophase, which is the last phase of mitosis. And this is where that cell elongation is continuing. The daughter nuclei are going to begin to form at opposite ends of the cell. So at each pole, a new nucleus is going to start forming. The spindle fibers are going to start disappearing. The chromatin is going to begin uncoiling again. And then cytokinesis is going to occur. Now, cytokinesis is not technically part of telophase, but it happens at the same time. and cytokinesis is essentially the division of the cytoplasm. So cytokinesis is going to occur a little bit differently in animal cells and in plant cells. In animal cells, it's pretty simple. Cytokinesis occurs through cleavage. And what happens during cleavage is just that a cleavage furrow is going to form, which is kind of like a shallow groove in the cell surface that you can see under a microscope. And then in the area that cleavage furrow forms is a ring of actin microfilaments that will then interact with some myosin, and then the cell is basically going to be pinched in two. And when that's completed, voila, you get two lovely little daughter cells. Now, in plant cells, cytokinesis is a little bit more complicated because they have that lovely cell wall to be concerned with. Well, to deal with this, what happens is sort of during those phases in that middle part there, you're going to get these vesicles containing cell wall material that's going to start collecting in the middle of the cell. And what's known as the cell plate is going to form. And once the cell plate forms, it's going to continue growing from the middle outward to the two ends. And then the membrane of the cell plate is going to fuse with the membrane of that parent cell. And then in between the two membranes of that cell plate, you're going to get new wall forming, new wall growing that's eventually going to reach either end. And then you're going to end up with two daughter plant cells. So it's just a little bit more complicated. There's not a pinching off like in the animal cell. It's more of a cutting from the middle outwards, whereas the animal cell is more of a slicing inwards. So what exactly controls cell division? Well, the answer to that question's a little bit complicated because it's going to be a little bit different no matter what type of cell you're looking at. But in general, we have a few things that are pretty common amongst different types of cells and different types of tissues and different types of organisms. One thing that has been noted, especially in animal cells, is anchorage dependence. And basically what this means is that in order to divide, the cell needs to be in contact with a solid surface. So in the organism, it would be that extracellular matrix of those tissues. So the cell would be in contact with that solid tissue, it would be able to divide. In a lab setting, it's usually like the bottom of the petri dish. And there's also another thing that's very important that keeps cells from kind of growing out of control is what's known as density-dependent inhibition. So when cells get too crowded, they'll stop dividing. This has been seen a lot in things like petri dishes, where usually a single layer of cells will form and then they'll kind of stop. There'll be too many cells, they're not going to want to divide anymore. But you can remove a few cells, and then the ones that are no longer bound in by cells on that side will begin to divide again, and then rinse, repeat. And how exactly do cells kind of know when to divide? What kind of signals them to do that? Well, we have proteins known as growth factors that are going to support cell division and help tell the cell when to divide. And then different growth factors and different combinations of growth factors are going to signal different kinds of cells when to divide and when to stop dividing, so on and so forth. And all of this kind of feeds into a cell cycle control system, which is basically the system that triggers and coordinates the events in the cell cycle. And pretty much what it is, what we're seeing here in this diagram, is that it's just like a system of checkpoints that stop the cell cycle unless they're overridden by signals from specific growth factors. And so basically, if the cell doesn't get the signal at the particular checkpoint, the division stops and it just doesn't happen. The big important one, though, is going to be the end of G1. There's this G1 checkpoint, and if the cell never receives the signal to continue on with the DNA synthesis, it's going to stop dividing. That cell lineage is going to eventually die out, and the cell will enter what is known as G0 phase. So the cell is just going to carry out its life cycle till it dies, and that's pretty much the end. Now, in normal circumstances, cells are not going to divide indefinitely. They're not going to be immortal. They're going to reach a certain number of divisions and then stop, because there's always going to be younger cells that will keep dividing and carrying on. But what happens when that doesn't get inhibited? What happens when cells begin to divide in an uncontrolled manner? Well, what happens then is usually cells that begin to divide without regulation or have some sort of abnormal reproduction like that, they're usually destroyed by the immune system. However, when that doesn't happen and they evade destruction, they can continue to divide out of control and form a mass or a lump known as a tumor. There are two main types of tumors. The first being a benign tumor, which is when the cells that are kind of dividing out of control just remain in that original mass or lump, and they don't spread anywhere else. Now, benign tumors can still cause problems depending on where they are, but they are not considered, for the most part, to be immediate dangers to the organism that has them. But again, depending on where they are, they can cause problems, especially if they are in the brain or an area that they're going to overcrowd. The other kind of tumor is a malignant tumor. And this is where these abnormally reproducing cells invade other tissues and other parts of the body. If you have malignant tumors, they are cancerous and you are said to have cancer. And then when these cancer cells spread beyond their original state, beyond that original little lump, they reach what is known as metastasis. So when you have these cells in metastasis, they're invading different parts of the body, they have spread, and the cancer is much more serious. That's as far as we're really going to go in depth in that. The book does have a little bit more in depth on cancer and tumors, but I don't study that particular area of biology very much, so I don't want to say something that's not entirely accurate. And generally in an entry-level bio class, the important thing is just to know the difference between a benign and a malignant tumor. So remember, a benign tumor is when those uncontrollably dividing cells remain in the area of the original mass or lump and don't spread to the surrounding tissues. A malignant tumor is when those cells kind of escape that original area and spread to the surrounding tissues and can spread throughout the body as well. Malignant tumors are considered cancerous, benign tumors are not. So that's all I really have for you guys this week. Thanks for kind of sticking in there for this whole video. I know this is kind of a long one, but cell division's cool and mitosis is pretty cool. Next video, we're going to go into meiosis, which is similar to mitosis, but different. So I hope you have a wonderful rest of your day, afternoon, evening, what have you. I apologize for the ducks yelling in the background, and I will see you all in the next one. Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane V. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.